we got to review a couple of things while you're getting signed in that are going to have to do a bit with this activity, plus they were the important things that we covered kind of right before our week off. Okay, the three things that we want to cover are the big three in terms of cellular transport. Okay, we talked about osmosis before the break. Okay, and we got to remember that osmosis is the movement of only water. Okay, that's why it's got a star beside it. Osmosis only moves water, and it moves it across the membrane. Okay, and it does that in order to balance solutes or salts. Okay, their concentrations, and it is passive. It is not something that the cell can control. Okay, it just happens. So if I put a cell in distilled water, which way is the water going to move? If it's going to try and balance salt concentrations, it's right. It's going to go into the cell, and eventually the cell will what? It'll explode. There's no way to achieve a balance there. There's no salts in distilled water. So the water will just continue to gush through the membrane. Okay, the cell will swell and eventually burst. Okay, the exact opposite would happen if I was to put a cell in heavily concentrated salt water. Okay, water would then rush out of the cell in order to try and balance all the salt that is outside of it. Of course, there's not enough water inside a single cell to do that. Okay, so the cell would just shrivel up. Okay, that's osmosis. The cell cannot control that. It is the passive movement of water across semi-permeable cell membrane. Okay, the other one that we said was really important is diffusion. Okay, and that is because diffusion moves anything, and it moves it within the cell. In fact, it is the only way material is transported within the cell. What's the fluid that it transports through? Cytoplasm, right? Okay, diffusion occurs through the cytoplasm. That's the fluid that fills up the cell, okay, and facilitates diffusion. Okay, it's also passive. Okay, it'll naturally move things from high concentrations to low concentrations, which generally gets them where they need to go. Okay, generally where they're in low concentration is where they're needed, so this stuff will generally make that work. Okay, and I keep saying generally because it isn't always 100% accurate, okay? This is not, okay, like, you know, Canada Post, where if you address something, it actually most of the time gets there, okay? This is well, if I make a whole bunch of copies of that letter and throw them into the air, one of them might get there if the wind is right, okay? It's much more of a shotgun approach to delivery, okay? So it moves from high to low. It's slow, and it's not targeted. That's what this chicken scratch down here says, okay? Not targeted. I cannot make by diffusion something go to a specific place. I can make it go in general to an area where there's very little of that material. Everybody follow me there? Okay, what's one of the other big drawbacks of diffusion? Right, it only works good across short distances, like the inside of a cell. Cells are small. They're small for a reason. Okay, they're small because diffusion doesn't work if they get too big. That's the purpose of today's activity, is to kind of illustrate how quickly a cell gets too big for diffusion to be an accurate way or a reliable way or an efficient way to transport things. Okay, the other method of cell transport is active transport. This activity has nothing to do with active transport, okay, but we want to review it anyway. Okay, it's different from these two in that it is active and thus uses energy. If a cell is going to use energy to move stuff across a membrane, it needs it pretty bad. All right, so it's going to use energy. It's going to move it against the gradient. That means it can pull it from an area of low concentration to high concentration. It's like pumping water uphill. Okay, it's not just water though. Basically, it could be anything. All right, and it does that again across the membrane. So there's those key points, those key identifiers we need to remember, because on a unit exam, I could give you a situational question and say, which cell transport method is going to be most involved in this situation? Explain how it'll work and why. Okay, everybody follow there. All right. Okay, you might want to copy this down somewhere. It isn't in your notes, okay, but it's valuable in terms of doing your lab report for this, but it's also something that ends up on tests. Okay, so cells absorb nutrients and they excrete wastes through their membrane. Okay, that's the only way a cell can get stuff in or get stuff out, is across the membrane. Okay? It's not as complex as we are, where we exchange gases with our lungs, we exchange material, okay, through our digestive tract, one end for intake, one end for output, okay? Things like that. There's much. We also excrete fluid wastes from a different place. We have way different ways of getting rid of and intaking material. Okay? A cell has only one way, the membrane. It can get in through the membrane, it can get out through the membrane. That's it. The paragraph, the part that I moved up too high. Okay, and that is their kind of outer surface area. It's almost, not completely, but almost two-dimensional. Okay, it's this surface area that covers the entire cell. That's the only way stuff gets in, it's the only way stuff gets out. So would it be good to have lots of it? Do you think? Would lots of membrane be good? Actually, yes. The more membrane you have, the better, because that is the only place where you can take stuff in, right? If you were in an eating competition, okay, having a big mouth would probably be advantageous. If you're one of those people that eats hot dogs for sport, it's disgusting, but people do it, okay? Having a big mouth is a huge advantage, okay? Because you can shove more hot dogs in there more quickly. Maybe you can just open your throat and just jam them straight into your stomach. I don't know. It's disgusting either way. Okay, but yeah, having a large area to take stuff in is a good idea. Imagine if you're moving into a house. Would it be easier to carry everything in through a big garage door or through a screen door? A garage door is a bigger opening. Okay, that's what we're looking at here. The more membrane we have, the easier it is, and the more surface area we have to acquire things and get rid of things. The problem is that as a cell grows, not only does its surface area increase, but its volume inside increases as well. Okay? The volume is a concern. The volume of a cell is where diffusion has to be responsible for moving stuff. The bigger the volume gets, the slower diffusion is going to be at moving material, because the distance is increasing. Okay, so it is because of the limitations of diffusion, that is its speed and that it's not targeted, that cells cannot reach excessive sizes. All right? At least not on all dimensions, because a cell is three-dimensional. Okay? Its membrane we envision as kind of a two-dimensional layer, okay? But the cell itself has length, width, and depth. Okay? So a cell can't get very big because it would take too long to move materials around. So imagine I've got these two islands, let's say. Okay? They're cells. We're going to represent a cell with an island. Okay? One island is very big. The other island is very small. The only way to get raw materials is for them to be brought in by boat. Okay? So raw materials can come to any point. They don't have a loading dock. They can come to any point on the outside of the island. Then those raw materials can only be moved by people carrying them from the shore to wherever they need to go. Okay? Which island is that going to happen faster on, the small island or the big island? The small island, because they don't have to carry it as far. The island is smaller. Okay? Now, the other problem is that not only do I have to, on the big island, transport raw materials a fairly long way to get to where they need to go, but whatever I do with those raw materials also has to be transported from one point within the island to another. So let's say that this is like a blacksmith, okay? And they're responsible for making things that are made out of metal, all right? Well, over here is where the stables are for the horses. So the blacksmith gets his steel from wherever on the island it comes in. Maybe it's over here and it's got to go all the way across. He makes the horseshoes, and then they have to be transported by carrying them all the way across the island to where the horses are. Okay? Whereas in the small cell, the steel only has to go to the blacksmith and then around here to where the stable is, not nearly as far. Okay? So essentially what I'm saying is a cell will die if it doesn't get what it needs quickly. So if I'm a really big cell, I might die before certain parts of me get what they need in order to keep me alive. To carry out those basic functions of the organism, they need certain things. And if those are not transported quickly, accurately, or reliably, the cell dies. Okay? That is why there isn't a giant amoeba sitting in a desk in here. Okay, it wouldn't transport materials effectively enough because it doesn't have a circulatory system like we do. Okay, as you get bigger, you still keep small cells, but they're supplied by a more active system of transportation, like our circulatory system. Okay, that's the only way you can get bigger, is to have other ways of transporting material that are active. Okay, everyone follow me there. A single cell could not be as big as we are and survive, because it could take hours for something to diffuse from one end of it to another. So imagine you ate a meal, and it took hours for the energy within that meal, okay, to be transported from the point of digestion to the rest of your body. Okay? Well, I mean, for us, the process of digestion takes about an hour, and then the energy we get from that can be transported within seconds. Okay, from the intestine to the rest of your body. Okay, we have an adaptation, okay, that allows us to be big. Everyone follow me on that? Okay, so as the cell gets bigger, it's not just that there's more surface area, which was good, it's that the volume, the interior volume of the cell gets bigger even faster than that, because this is three-dimensional. The surface area is only two-dimensional. So how do I calculate surface area? Anybody remember? If I have a cube, how do I calculate surface area? That's volume. Length times width times height is volume, okay? Yeah, to get the surface area, if I had a cube, how would I do it? Length times width times 6 times the number of sides, right? That would give me my surface area of a cube. Well, if I make a cube get bigger, 2 of its parts get bigger. The 6 stays the same, okay? So if I go from having a cube that's 10 by 10 then on a side, and then I go to 100 by 100, it only gets so much bigger. But if I'm looking at that same cell's increase in volume, all 3 dimensions get bigger. So the number gets bigger faster. Okay, it would be, instead of 10 times 10 times 10, it would be 100 times 100 times 100, instead of 100 times 100 times 6. Okay, so volume increases faster than surface area as a cell grows. Okay, and what happens is the cell's surface area can't supply the greater volume with materials fast enough, and diffusion can't move through the increased volume fast enough, and then a cell becomes inefficient and dies. Okay, or a cell gets smart. What else can a cell do as it gets bigger? Divide. Okay, and there's usually an upper limit to size where a cell finally goes, you know what, it's just too hard, I'm going to split it. Okay, and they do. Okay, you split in half, now you got 2 smaller cells. Okay, that's how it kind of goes. All right, so our surface area to volume activity is going to look like this. You've got a Google Google Doc, okay, that you're going to be filling in this stuff on. There's also a PDF that looks like this sheet that I've attached to the assignment. Okay, so the problem we're investigating is, how does the surface area relate to the volume in cells of different sizes? And we've kind of already talked about that. Does dividing a large cell into smaller cells affect the surface area to volume ratio? What we want to look at is, how much surface area to volume do we have? Okay, which number do we want to be bigger? Surface area or volume? Surface area. Okay, we want big surface area for acquisition of materials and excretion of waste, and a small volume to transport all that stuff across. Okay, so if surface area is big and volume is small, the cell is good. If volume is big and surface area is small, the cell's in trouble. Okay, so that's what we need to look at there. Okay, so for our hypothesis, you need to generate a hypothesis about the relationship between surface area and volume in cells, and how that can affect the functioning of a cell, and I would say specifically talk about transport by diffusion here. Okay, as a cell gets bigger, what's going to happen to its ability to transport by diffusion? So what we're going to do is we're going to use these 3 imaginary organisms: micro, macro, and multi-micro. They're 3 different organisms. Okay, micro is a single-celled organism that is very small. Macro is a single-celled organism that is very large. Multi-micro is a multicellular organism that is the same size as this. Okay, everybody with me there? All right, so what we want to do is look at their surface areas and their volumes and compare them. Okay, what's happening as a cell gets bigger? Okay, And does making a cell multicellular make the transport situation better by changing the ratio of surface area to volume? All right. Obviously, it must. We're multicellular and we're big. Okay. We don't seem to have a problem diffusing material, transporting material. Okay. Once we've divided into lots and lots of small cells. Okay. So we got to do a little bit of math. There's a chart in there that we're going to fill in together. Okay. Do the math together here and save us a little bit of time. Okay. So your chart looks something like this, except neater. It's not hand scrawled like this one. Okay. So for surface area for micro. Okay, micro's dimensions are that it is 1 micrometer by 1 micrometer by 1 micrometer. Okay, so it should be in your Google Doc. It's in the Google Doc. Is it? Oh, okay. So micro is 1 micrometer by 1 micrometer by 1 micrometer. So to calculate its surface area, we just said we go length times width times the number of sides. Because it's a cube, all the sides are equal. We can do that. Okay. All right. So for that one then, okay, that's going to be 1 times 6. There's 6 sides to a cube. So the surface area of micro is 6 square micrometers. Yes, I'm giving you the answers to the chart, which is worth 5 marks in your lab. So yeah. How do you get the square? You go make it, put a 2 there, highlight it, go to Tools, and in there should be superscript. Or Control comma does superscript. Okay, Control comma is the shortcut. So highlight it and go Control comma, and it should superscript. It's not. Oh, that's subscript. So superscript is Control period superscripts? Okay, Control period superscripts. Okay, to calculate the volume of micro, I go length times width times height. Well, it's 1 by 1 by 1. What's 1 times 1 times 1? 1. Okay, that's easy. 1 micrometer, so that's mu m cubed. Okay, 1 cubic micrometer. So our surface area to volume ratio is 6 to 1. Surface area is 6 times bigger than volume. Is that good? Yes. Yes. Okay, that is definitely a situation that would be desirable for a cell. Okay, to have a large surface area and a small volume by comparison. All right, for macro. Macro is 10 times bigger on each side than micro is, so it's much bigger. All right, so to calculate its surface area, I'm going to go 10 times 10, length times width, that's 100, times the number of sides, okay, which gives me 600 square micrometers. Calculate the volume, length times width times height. 10 times 10 times 10 is 1,000. All right, so look at what happened here. I increased each side by a factor of 10, and my volume increased by a factor of 1,000. My surface area only increased by a factor of 100. Okay, so as we get bigger, as a cell gets bigger, volume increases faster, and that's bad. Okay. This surface area to volume ratio is now 0.6 to 1. Which is bigger, volume or surface area? Volume. And we said that situation is not good. 0.6 because it's 600 to 1,000. If I divide each by 1,000, I get 0.6 to 1. Okay, that's the lowest terms for that ratio. Okay. Okay, now if I look at multi-micro, the box for multi-micro is the same size as macro, but it's made up of tiny little micros. Okay, how many little micros do you suppose are inside of there? Well, on each side there's 10, right? So if I filled up this whole thing with micros, there would be 1,000 of them. Okay, there'd be 1,000 micros inside of there. The surface area of each micro is 6 square micrometers. So the surface area for a multi-micro is 1,000 times greater than that, which is 6,000. Okay, now usually at this point people say to me, but Coder, a bunch of those are inside. They're not on the outside, doesn't count for the surface area. Well, you got a whole bunch of cells that are on the inside of you. Okay, does it make any difference? No, the surface area still counts. Okay, give you some idea, guys. If I was to take just your intestinal tract, your small intestine, in fact, okay, it's quite long, but there's also an incredible amount of surface area in there because it's got all these little folds inside for absorbing material that you eat. There's enough surface area in just your small intestine, and if I laid it out, and this is kind of gross, but I wouldn't do this obviously, okay, if I laid it out, it would cover a tennis court. There's that much surface area for absorbing food in your small intestine, more than a tennis court. Okay, it's a lot. Okay, it's a lot of surface area. Okay, now volume-wise, is it still the same size? It's still the same size as macro, right? So it still has the same volume. So the volume is still 1,000 cubic micrometers. That doesn't change. But did the surface area to volume ratio change? It did. Okay, now we're back to 6:1, which makes sense. If you're made up of a whole bunch of small cells, your surface area should be the same. Surface area to volume ratio, sorry, should be the same as each individual cell in your body. Okay, so becoming multicellular seems to overcome this problem. Okay, if you become big as a single cell, you're not going to function. But if you break yourself up into a whole bunch of little cells, you probably still work. Okay, that's what that chart is kind of saying to you. Okay, now a bunch of the analysis questions have to do with this chart. Okay, so for question number 1, it says when the cube dimensions increase from 1, it's supposed to be micro, it says centimeters here, from 1 to 10, so when you go from micro to macro, the increase for each side is a factor of 10. How much does the surface area increase? Okay, so basically they're saying, how many times bigger is the surface area of macro than micro for this question? And how many times bigger is the volume of macro than micro? So basically just a little bit of math for you to do there. Okay, number 2 is a similar question, except now they're comparing the two big ones. Okay, how many times greater is the surface area of multi-micro than macro? What about the volume? Okay, calculate the factor difference. So how many times greater? Okay, for number 3, for which two cubes does the ratio between the volume and surface area change when the size increases? So they're saying, if I make this one bigger, if I make micro bigger, will its surface area to volume ratio change? If I make this one bigger, will its surface area to volume ratio change? If I make this one bigger, will its surface area to volume ratio change? Now, keep in mind, if you make this one bigger, what does it fill the new space with? More little cells. Okay? So keep that in mind. Okay, and then for number 4, okay, you're looking at this diagram right here, okay, with these different shapes. What number 4 is asking about is, does the shape of a cell influence its surface area to volume ratio? Okay, so does its shape affect that? So what we got to look at here, okay, are the dimensions of these cells. Okay, now to calculate surface area and volume of these, you can't just go length times width times the number of sides, because only A is a cube. And it doesn't really look like a cube, but that's because I accidentally stretched the picture. It's supposed to be a cube. Okay, B is most obviously not a cube. Same with C. All right, so to calculate the surface areas of these shapes, I need to calculate for B the surface area of this side, and then multiply that by 2, because the back would be the same. Okay, find the surface area of this side, multiply it by 2, because over here would be the same. Find the surface area of the top, multiply it by 2, because the bottom is the same. Add those numbers together. That'll be the surface area of B. You got to do the same thing with C. Okay, and C, and then see if that changes the surface area to volume ratio. Because believe it or not, the volumes of these three are pretty much the same. When you go length times width times height for these three shapes, you're going to get very close to the same number. Okay? They are essentially the same volume. Right? What we need to see is, does changing their shape affect their surface area? Is that a way for cells to adapt? Okay, because some cells in your body, like the next question, okay, have different jobs, and as a result, need to have certain shapes. Okay, a nerve cell, for example, can be up to 1 meter long. Okay, the ones that run from the base of your brain to the small of your back through your spinal cord, okay, can be up to 1 meter long. That seems kind of big for a cell, but remember, they're only big in one dimension. Okay, their length, as long as they're really skinny, they might still be okay. We'll find out when we do the math for question number 4. Then we can use the math from number 4 to answer number 5. Okay? Everybody follow me on that. So the cells we're looking at in number 5 are a liver cell. Well, in your liver, you got little tiny cells, and they're all packed in there. But there's also tons and tons of blood vessels in your liver. Okay? Your liver holds more blood than any other organ in your body. Right? So they're always kind of well supplied. Their shape isn't really all that important. Okay? Skin cells is the other one. Well, they kind of face the outside. They can really only absorb stuff through 1 or 2 or maybe 3 faces of the cell. Okay? They're not absorbing anything through the top. Okay? We don't breathe through our skin or absorb food through our skin. Okay? So they don't do much that way. Okay, for question number 6, it's talking about the shape of a red blood cell. Okay? And you can see that a red blood cell is biconvex. Okay? What that means is it looks, if I'm looking at it from the side, it goes in and it's kind of hourglass shaped. Okay? So it looks like this. Is this shape better than just being perfectly round or perfectly spherical? Okay? Which one would have more surface area? Being perfectly spherical or indenting in to change your shape and see if you have. Okay, that's kind of for you to figure out there. You can give me an answer to that question. Okay, and then for number 7 is the most important question in the analysis. Okay, most materials get from place to place in the cell by diffusion. What effect would a low surface area to volume ratio have on intracellular transport? Intracellular means within the cell. Okay, we've talked about that already several times today, as well as before the break. Okay, so I want a good explanatory answer for number 7. Then you got your conclusion. There's no evaluation on this one because we didn't do anything. Okay? There wasn't an actual lab procedure, so there's no sources of error. Okay? This is simply a mathematical exercise. Okay? Are we following what is expected here? Okay. You still have 45 minutes. You should be able to get pretty close to done this whole thing, I would think, in that time. Okay. Now, for measurements, it's going to be pretty hard to measure these on the computer screen. Okay, I will measure them out. I thought I had them on here, but I will give you the dimensions of these. Okay, and then you guys have got to do the math for them. Okay, so you can do the other stuff, and then by the time you get to there, I'll have those ready. Okay, so your dimensions for question number 4. Box A is macro. It's 10 by 10 by 10. Okay, 10 millimeters by 10 millimeters by 10 millimeters. Okay, it's the same dimensions. It doesn't look like a cube, but when you put a ruler on it, it's a cube. It's 10 by 10 by 10. I don't know why. Okay, for B, okay, box B is 5 millimeters along this short side, 10 millimeters tall, and 20 millimeters long. Okay, and box C is 1 millimeter wide, okay, 10 millimeters tall, and 90 millimeters long. Okay, so those are your dimensions for calculating surface area and volume for question number 4. Okay, because it's pretty hard to measure them on a screen.